Hello everyone, welcome to the channel Microbial World. In this video, we will be learning about crown gall disease. What is crown gall disease? It is a plant disease causing tumor-like growths or galls mostly at the crown region, that is, the junction of root and stem. So, crown gall disease causes tumor-like swellings, or it is also called as galls, and these tumor-like swellings are usually seen at the crown region of the plants, which means the base of the stem. The disease affects dicot plants like roses, grapes, apples, and stone fruits. The disease is caused by the bacteria Agrobacterium tumefaciens. It is a gram-negative, rod-shaped motile bacterium. Agrobacterium tumefaciens is called a natural genetic engineer. This is because it transfers a piece of its DNA called tDNA into the plant genome. The transfer of tDNA is done through a Ti plasmid or tumor-inducing plasmid. This is the first natural example of horizontal gene transfer from bacteria to plants. Ti plasmid is a very large plasmid that is found in Agrobacterium tumefaciens, and it is responsible for causing crown gall disease. Ti plasmid contains several key functional regions such as tDNA region, opine catabolism genes, virulence genes, and origin of replication. The tDNA region contains genes for tumor formation or gall formation. It also contains genes for opine synthesis. Virulence genes is found outside tDNA. It helps in processing of tDNA, and it also helps to transfer tDNA into the plant cell. Opine catabolism genes allows bacteria to utilize opines. Opines are special amino acids that is used by bacteria as food. Origin of replication allows the plasmid to replicate inside the bacterial cell. Now let us look into the disease cycle of Agrobacterium tumefaciens. Agrobacterium tumefaciens is a soil-borne bacterium. So it survives in the rhizosphere, which is the root zone, or in the plant debris as a saprophyte, means it feeds on dead organic matter. Infection begins when the bacterium enters the plant through fresh wounds caused by insects, mechanical damage, or cultivation tools. The wounds release plant phenolic compounds like acetosyringone, which attracts the bacterium. The bacterium then attaches to the plant cells. It uses cell surface proteins to attach to the plant's wounded cells. This is a specific interaction, especially in dicotyledonous plants. Phenolic compounds activates the virulence genes on the Ti plasmid. These genes are responsible for DNA transfer machinery. A portion of the Ti plasmid called the tDNA or transfer DNA is then cut and transferred to the plant cell. The tDNA is then integrated into the plant genome. As we know, tDNA contains genes for gall formation. So it stimulates uncontrolled cell division and results in the tumor formation. tDNA also contains genes for opine synthesis. So it produces opines that is used by the bacterium as nutrients. The gall typically forms at the crown region, which is the root-stem junction, but it can also appear on the roots and stems. The bacterium multiplies in the gall tissue, and it can also spread into the soil and infect other wounded plants. As the infected plant dies or is pruned, the bacterium re-enters the soil and survives to restart the cycle. This is the diagram showing the disease cycle of Agrobacterium tumefaciens. As we already said, the bacterium survives in the soil as a saprophyte. The bacterium enters through the plant's wounded cells and it attaches to the plant cells. tDNA from the Ti plasmid is then transferred into the plant cell. The tDNA integrates into the plant genome, causing transformation. It causes uncontrolled cell division and results in gall formation or tumor formation. Young tumors are easily injured or attacked by secondary invaders. The bacteria from the gall surface moves into the soil when the tumor breaks. It survives in the soil and restarts the cycle. Symptoms of crown gall disease includes formation of irregular galls at the crown region, roots or stems. The galls are initially soft and white, but later it becomes hard, brown and woody. It results in stunted growth of the plants and yellowing of leaves. It also causes reduced flowering or fruiting. In severe cases, it can result in wilting and plant death. Control measures. Use disease-free planting materials. This means we always have to choose healthy gall-free seedlings or rootstocks and avoid using cuttings from infected plants. Now, since the bacterium is entering through plant wounds, we have to avoid causing injuries to the plants. So we have to handle the plants very carefully during pruning, grafting or transplanting, and also disinfect the tools between uses. Remove and burn any infected plants. Another measure is to do crop rotation with non-host crops like grasses or cereals to reduce the soil inoculum levels. Apply biological control by using Agrobacterium radiobacter strain K84. It is a naturally occurring non-pathogenic bacterium. It produces agrocin 84, which inhibits Agrobacterium tumefaciens. Maintain field hygiene and disinfect tools. Avoid overwatering because excess moisture can favor bacterial growth and increase root wounds. Also ensure good drainage. So this is all about crown gall disease. I hope that the video was useful for everyone. Do comment your doubts and subscribe the channel for more informative videos. Thank you for watching.